Hello and welcome everybody to the September edition of our PathSafe webinar series. Today we're going to be joined by colleagues from the Animal and Plant Health Agency, APHA, National Milk Records, NMR and the Agricultural Industries Confederation, AIC, who are all involved in delivering two of the PathSafe workstream 2B AMR Surveillance Pilot projects, that is the dairy cattle and the animal feed surveys. So we'll start with an introduction and overview of the projects from Chris Teal, Head of Antimicrobial Resistance at APHA. Then we'll have Eamon Watson, Product Strategy Manager at NMR, and Simon Williams, Technical Manager at AIC, who will provide some context and background from their organisation's perspectives of participating in the programme. And then last but not least, Manal Abu-Owen, Senior Scientific Officer at APHA, will share some insights into the lab logistics and coordination aspects of these projects. And then following the presentations, we will have a Q&A session. So before we kick off, just a few quick housekeeping elements. First of all, the session today is going to be recorded, and the recording, along with the slides, will be saved on the PathSafe webpage in due course. The session should run for an hour, so we'll be finishing at 3 p.m. And if you could all please keep your microphones on mute if you're not speaking, that would be greatly appreciated. As I said a second ago, we will have a Q&A session following the presentations, so if you feel free to use the Q&A function to post any questions you have. And then when we come to the Q&A at the end, we'll ask you to unmute and ask your question. And any questions that we don't get to or can't be answered during the meeting, we'll try and follow up with offline afterwards. And because we've activated the Q&A function, the chat function has been disabled. So there isn't a chat option, but there is this Q&A function that you can use. And I think Rachel is going to pin a link to the PathSafe webpage in the Q&A section for your reference. So hopefully that's it. Rachel will remind me if I've missed anything. Failing that, I'll now hand over to Chris, please, to start us off.
Thanks very much, Ed. And I'm going to talk about two projects which we were very pleased to be involved with. Surveillance of national milk laboratory bulk milk, and also a study of animal feed. So next slide, please. Thanks. So if we think about the EU surveillance that's done, it covers 4 major sort of sectors and species. So fattening turkeys, fattening pigs, broilers, and veal calves. So obviously UK doesn't have a big veal industry, but we do have quite a big dairy industry and a big beef industry. And those aren't covered by the statutory EU surveillance. So one of the things we're interested in is how can dairy cattle be surveyed on a national basis? And of course, we already have bovine bulk milk, which is collected and tested for various purposes, including some statutory purposes. So things like checking for brucella, freedom of brucella for the UK, that sort of thing. It's also used for other purposes. So investigating the occurrence of bacterial mastitis pathogens, other organisms. And with a sort of raft of different purposes, things are done on it. So we've got total viable counts, hygienic milk production, measures of mastitis, milk quality, residue testings done for food safety. And there's also the sort of cattle health basics, cattle health possibilities for using on bulk milk, so standard bacteriology, but also serology and exposure to various diseases and conditions. Then from the public health perspective, raw milk, it can be consumed in parts of the UK. It's not in all parts, I don't think, but and also, you know, there are some unusual uses. So one thing which came up in some of our discussions is that, you know, it is offered for sale as bathing milk in the way that Cleopatra would have used it. So some maybe unusual uses that don't spring to mind at first sight. So we've got this great opportunity to look at bulk milk and see how a collector look at a whole load of samples from across GB in collaboration with national milk laboratories, and to try to integrate that with other parallel PathSafe studies. The herds are anonymized, so we can't get all of the data that we might perhaps like, but we still think it's a good opportunity. I hope you can see the next slide now. This is number 4. But if we're thinking about the individual cattle herds, so we've got the animals and we've got antimicrobial usage in different herds at different extents. We've got animal movements on and off herds which aren't closed. And then we've got exposure of the cattle to antibiotic resistant organisms in the environment, in their feed or water, or through other contacts, including their human contacts. So we've got a whole load of things which are influencing what's going on in the cows. And then a whole load of other things affect what eventually turns up in bacteria in the bacteria in bulk milk. So if we think about bulk milk, we've got sources of bacteria from the cow's environment, its bedding areas, loafing areas, feed pastures. We've got mastitis pathogens, specific organisms which are infecting the udder. Then we've got some bacteria which are coming from the milking plant. So dairy hygiene is quite a significant thing in influencing how those occur. Then at the end of the sort of production line, the milk's refrigerated while it's awaiting collection. So that's also a sort of an opportunity for some types of bacteria. They quite like that cold environment where they are. Their growth is favoured. So if we think about the inputs that are occurring in the cows, then that's quite a big variable. So there'll be big differences between different herds, how much antibiotics used, for example. Then once the milk leaves the cow from the cow's udder, we've got a whole raft of processes which are designed to be put in place, which control how many bacteria are going into milk. So those control processes could be a factor which smooth the variability between the individual dairy cattle herds. What I mean is you might have one herd that has a lot of resistance, one herd with not much resistance, one herd has high levels of bacteria in milk, another herd has low levels. But as it leaves the cow into those first sort of pipelines of the milking system, but then there are a whole raft of other procedures which influence what's actually turning up in the bulk milk. So those might smooth the variability between the herds. So that was one of the things that we wanted to look at. Also, I mean, it's fairly well established that the optimal gram-negative and gram-positive indicator organisms, if you're thinking about the intestine of domestic animals, feed-producing animals, are pretty much across the board. Now, I think E. coli and enterococci are used. But are these the best things to look at for in bulk milk? Because there's different survival of different members of the Enterobacteriales, for example. So although we're picking E. coli often when we're looking at intestinal work, might other organisms be more appropriate here? Things like Klebsiella pneumoniae. Also, some of the mastitis pathogens, so Staph aureus, and because it's the occurrence of MRSA, development of methicillin resistance. So that's another organism that it's interesting to look at in milk. If we think about the sort of traditional bacteriology approaches, we've also put into this project, we wanted to try and find a general indicator of the degree of resistance in all the bacteria that are present in milk, or most of the bacteria, at least the bacteria that can carry class 1 integrons. So class 1 integrons are genetic packages with which resistance is really frequently associated. So that part of the work involves developing a quantitative PCR to look for the occurrence of those class 1 integrons. And that's where we think there might be quite big differences between herds and the level of occurrence of those between different herds. But that's what's under investigation. So to sort of summarize the milk study, so we've got the scale of differences between herds, which is reflected in bulk milk. We can look at size of the herd. We can imply the size of the herd because we know from NML, Eamon and colleagues, how much milk was collected. We also know which region the milk came from, and we can look for associations between region and what we see in the bulk milk. So with that, I suppose, you know, all herds across the GB would be different in terms of their antimicrobial usage, and there might be regional trends in the amount of usage. But one thing which might also influence the regional AMR associations is what's in the environment and what the cows get exposed to through their environment while they're at pasture. So grazing at pasture or grazing adjacent to watercourses, what they're exposed to could be really important in what sort of levels of resistance they finally get. We observe in their intestine, and we think about sort of discharges of raw sewage into rivers, that sort of thing. Are those important? In that case, that could be a factor which does influence what we can see. Also, the characterization of the isolates, so strain types and the what resistance are present in all the types of bacteria we might find. So the mastitis bacteria, the zoonotic and the commensal, and the other bacteria which we see in milk is of interest. And finally, the relation to those bacteria which occur in humans. So trying that will involve probably pulling all the strands of PathSafe together, because there are other strands of PathSafe which are looking more specifically at the types of bacteria and the strain types occurring in man and humans. Moving on now to the feed study. So feed is of interest because there are not so many previous studies that have been done on feed. A few studies in other countries, which have usually shown limited detection of AMR in the bacteria detected in feed. But of course, if resistant bacteria in feed, then there's the potential to affect multiple herds and flocks if the feed's distributed quite widely. And recent studies in Sweden and America have both commented that the possible role of feed in dissemination of AMR is not very well defined currently in the literature. So this is a collaboration with the Agricultural Industries Confederation. And one thing we wanted to look at was both the ingredients that sort of enter the process and the finished feed, because a lot of the feed production processes result in a reduction in bacterial viability. So, what actually the animals receive might be even have fewer bacteria than the raw ingredients that sort of enter the process. And of course, from the UK perspective, we know currently that there are multi-drug resistant salmonella affecting food-producing animals across the globe, really, and each region of the globe tends to have its own salmonellas, and they are quite prevalent in livestock in those sectors. So one of the things the UK it's very beneficial for the UK to do is to keep those out, because we know they have the potential to spread epidemically in food-producing animals, and they have done elsewhere. So that's another sort of strand to this. I mean, feed is quite intensively sampled already for salmonella, but it's another strand, and illustrates the importance maybe of how things might enter the country that they're going to have quite big implications if we don't take these measures to contain them. Last slide from me is lastly, to finish up, really the indicator organisms appropriate for feed. So we mentioned milk and how there are a wide range of bacteria in milk, and which bacteria are most important, which can we get most information from. And similar considerations really apply to feed. So the American study I mentioned, which was a feed and raw pet food, did target E. coli and the enterococci, so similar to what we'd look at in the gut of animals. But the Swedish study looked at other enterobacteriales as well, so Klebsiella, Enterobacter, and Rauwoltella, and they actually found something really interesting in an Enterobacter. So I think the point here is that those organisms have differential survival. If we think about Klebsiella, it's got a capsule. It survives really well in surface waters, much better than E. coli does. And that differential survival may mean that it's not really optimal to target one of them. We have to, across the board, we need to tailor our approach to what is persisting and surviving in that particular matrix. So in broad outline, the study is going to look at different types of bacteria and the occurrence of AMR in those bacteria in various types of feed materials, and both looking with non-selective and selective cultures. So that's a very brief introduction and overview, and I'll hand over to Eamon there. Thanks.
Okay, thanks, Chris. Good afternoon, everyone, and thank you, Rachel, Ed, and everyone at the PathSafe team for the opportunity to talk about our role as National Milk Records and why we've got involved with this project. Just very briefly, introduction from me, Eamon Watson, and I am a vet, and I've enjoyed a career with dairy cows, with laboratory diagnostics, and working with epidemiology and surveillance. And I joined National Milk Records about 8 years ago, which has been a sort of perfect blend of everything that's gone before. I'm currently a strategy manager, product strategy manager, which very much means I look to the future and keeping an eye on what's coming down the pipeline, just to ensure that products and services are fit for purpose in the years ahead. And so, you know, keeping a sort of active brief on these what might be peripheral to our core business projects are important, because of course what's coming down the pipeline will impact us all and across the whole dairy sector. So Ed, can I have the next slide, please? So who, for those of you who don't know, who are National Milk Records, and you know, why would we get involved in the PathSafe project? So as NMR, we're independent and we work right across the dairy sector. So we're open to working with all producers, all dairy farmers, all vets, all milk processors, and there's upwards of 90 processors, number of larger processors that will have 60-70% of the GB milk field. But there's 90 plus processors, and we'll work across with all of them. We're open to working with other bodies, including government agencies, university researchers, and wider dairy sector and industry. So we're very broad in who we work with, and we're very fortunate in having that sort of independent status and being able to dip in and work with everyone. We've three main arms to the business. The classic one that a lot of people will associate us with is the milk recording for producers. So that's the milk recorders, the NMR milk recorders going onto farm, collecting milk samples from cows from morning and evening milkings, and yield data, farm event data or herd event data. Bring the samples end up in our laboratories, and we test them through for cell counts and fat and protein status. And in terms of GB, we're representative. It's approximately 40% of herds, 50% of cows, and about 60% of the milk production. So it's slightly favoured towards the larger herds and the higher producing herds. So the second arm to our core business is for payment testing for the processors, for the milk buyers, and this is really where the PathSafe samples fit in. So these are the samples that will be collected as the tanker driver goes onto the farm. They'll go as well as collecting the bulk tank, the whole vat of milk. They'll collect an aliquot sample, a 40 ml pot of that sample, and all of those samples from all the farms that are collected across GB will end up in our laboratories, and we would do termed it there, payment testing. And the payment testing will be about the composition of the milk, the fat and protein, lactose, urea, fatty acid composition. We'll do hygienic status, so back to scan testing, maybe some bacteriology for TBCs on that as well. Thermodurics are another parameter of interest that's coming in with the milk processors. And we'll do testing for residues, so testing for antibiotic residues. So that's the 40 ml pot of milk that the milk tanker driver will collect from every collection from every dairy farm, and that ends up as a network of collection points, but they will all end up in our laboratories. So milk collected 3 o'clock this afternoon, we'll have that sample with the milk tanker be collected, and will be in our laboratory at midnight or 1 o'clock in the morning, and our night teams will test that overnight to enable results out to producers by 7-8 o'clock in the morning. The third arm for the business is very much related to the laboratories and the diagnostics that we provide. So that's health and disease testing, primarily for vets and their producer clients, but we will do testing programs on behalf of milk processors as well. So we have 2 laboratories, one in Wolverhampton, or just outside Wolverhampton, and one in just outside Glasgow. Both laboratories UCAS accredited testing. And we've got a wide range of tests from, I mentioned the bacteriology, but a lot of work on ELISA. I think as Chris mentioned there, the use of the milk samples, a lot of ELISA testing. Our herd-wise scheme for Johne's will rely on ELISA testing to test for Johne's disease. And we also provide genomic testing, so identifying the breeding potential of cattle. And most recently, or what's coming to the fore now, is GeneSells. I'll just mention it briefly because it's an interesting blend, and a sort of, you know, shows our desire to be ahead and lead the way in terms of what we're doing with the dairy industry. So GeneSells takes the genomic evaluations from the animals, so we know the DNA profiles of every cow that's in the herd, and we can now do milk. We can do cell counting based on a bulk milk sample. So instead of being reliant on individual cow samples, we can now take a bulk milk sample and do it through the genomics laboratory, and that will identify the DNA that's in all the cells in that bulk milk sample, which we can then attribute to a cell count. So quite an exciting development, quite a game changer in terms of that traditional cell counting approach. So you might see GeneSells sort of float to the fore. There's a few other parts to the business. I mentioned the Nordic Star tags. You can see the heifers in the picture below proudly displaying their tags, but of course they're primary identification tags and also the tissue sampling tags. So it's a big, we're across the board working with everyone, wide range of tests, keen to collaborate, very open to collaborate, and just recognize that there's potentially a bank of samples sitting in that fridge that are open for this sort of research and epidemiological purposes. Okay, next slide, please. So why NMR? What would be our interest in terms of antimicrobial resistance and PathSafe? One of the projects that we do is PharmAssist, and PharmAssist is it's been running for a number of years now, 5 or 6 years, and we work with milk processors and we gather in antimicrobial use records from vets for those processors producers. And so that enables it's a very sort of active approach to recording antibiotic use. And for those processors that have been on the scheme for some time, they're able, of course, then to both measure the antibiotic use across their herds and the change or the trend in that antimicrobial use, and also benchmark it to the wider data set, so all the data set that's in the PharmAssist service. So we're now into our 7th year of data collection. There's multiple processors. There's about 15 processors, retail groups that will be recording through PharmAssist. We're gathering data from 210, just over 210 vet practices. There's about 1,200 herds that we'll regularly or annually take in data from, covering about 350,000 cows. So that's PharmAssist has been a service that's been established and running for some time, and of course we've got that record of antimicrobial use, which sort of leads on to my sort of the second graphic in that slide, in terms of so what. But I've always just sort of harbored this interest in, okay, well, what's a farm's resistance profile and has it changed? And because we've been measuring that antimicrobial use over a number of years, we've seen a change in the level of use and also the pattern of use. So the graphic below, the syringes, that's the HPCIA injectable use over the last few years, and the red bar just illustrates the level it was at. But we can see, you know, that's fallen away, not quite to zero, but virtually to zero across herds. And it just sort of harbored that question. I wonder what the resistance would have been 5, 6 years, 10 years ago, compared to how it is now, or how it's going to be into the future. And there was no commercially available testing just for us to sort of go and explore that. We do have a PCR for mastitis pathogens, and within the PCR there's a one of the testing can be for the beta-lactamase resistance gene, but aside from that, you know, very limited on what we could do in terms of antimicrobial resistance gene, and certainly profiling of farms. So it was with a sort of open ear that we stumbled across PathSafe and were keen to get involved. So for us, you know, it's interesting. It's hugely relevant for the dairy sector. We recognise that there's a fridge full of samples. You know, there's a sample there from every farm, every collection, every day. And I put there opportunist epidemiology, which is really what this is. You know, there's a bank of samples. Can we work with partners and offer those where we can to be available for suitable research projects? So we all benefit from this. You know, we'll learn a little bit. Our processor colleagues will learn a little bit, and you know, the whole dairy industry, the whole dairy sector will learn from it. The third part, among the green box on the slide there. So just to highlight what is it that we're doing, what's our role in this. So the milk samples are the payment testing samples. So these are collected by the processor, and we do the testing on behalf of the processor. So it's important to acknowledge and recognize that we've got the permission of a processor to use those payment samples. So it's opportunist epidemiology, but where we have the permission to do so, and unfortunately we've got a good relationship with processors, so more often than not we're able to facilitate these sort of requests. So the protocol or the setup is that we identify 100 randomly, but they're proportional to the number of farms in each NUTS1 region across GB. So we identify 100 each month, and those samples will be selected at 25 per week, and they'll be processed by the laboratory team. And I just highlight their brilliant laboratory team. So they're very engaged. They're very keen to participate in this. They're very attentive to ensure that the samples are collected properly, are subdivided out, because we've got to provide two sub samples, and have been dispatched. So big thanks and acknowledgement to our lab team for pulling out the samples. So there's 100 samples a month, and we're scheduled to provide 1,000 samples, which will be done between that period of January and October. There's likely to be a few more, just because some of the herds will have multi-vat, so where they might have two milk tank collections, we will have taken samples from both of those milk vats. And that's really our involvement. We're sort of enabling the dip into the fridge to randomize across GB, proportional to NUTS1, and we're transferring those samples on to APHA and colleagues to do the testing from it. And I did check in with the lab team, and as best as I'm aware, no issues in terms of working with PathSafe project. I think there's been an occasional courier delay, but aside from that, there's been no issues whatsoever with collecting the samples or forwarding them on. Okay, and my final slide, please. And final slide, it's a bit of a plug, but for those that if you haven't seen the pharmacist or not aware of what pharmacist does, there is a summary report which is 5 years. It's now in preparation to bring it through with the data to 2022. So that'll be 6 years worth of data, and that short summary report is available on the NMR website. I think if the slides are being circulated, that title will be is the hyperlink to it. But if anyone has any difficulty, they can come to me direct, and I can signpost you to it. But that's an openly available. We do it every year, and just update. You see the charts on the right there. There's just a changing pattern of antimicrobial use that we have recorded in Pharmacist for those. Like I say, it's approximately 1,200 herds that will have regular medicine use data from. Okay, that's all I have. Happy to take questions at the end, and I am now going to pass over to Simon.
Thank you, Eamon. Good afternoon, everyone. My name's Simon Williams. I work for the AIC. Everyone else has said the full name enough, so I won't repeat that. And my role is I'm a technical manager. I look after the feed certification standards that AIC operate, and I think it's through that the FSA and VMD managed to track me down and invite us to be involved in the PathSafe project. So next slide, please, Rachel. So AIC is primarily a trade association, and we represent the agri-supply industry. So the area I work with is predominantly animal feed, but we also have sector specialists in crop protection and agronomy, fertilizer production and merchanting, grain and oilseed trading, and also seed production and trading. So we're, in that sense, we're quite a unique trade body in that we have multiple sectors. Most trade bodies tend to be very, very focused on one area, but we came together as a group 20 years ago, which is why we've got AIC 20-year logos, to sort of really ensure that the voice of the agri supply sector gets heard by policymakers. Next slide, please. So the structure of AIC is overall we've got an AIC group, then there's AIC, which is the trade association I just talked about, and then there's an affiliated organisation, the UK Former Foodstuff Processors, which has a big role in recycling food into animal feed. And then the organisation that I work for, which is AIC Services, which offers the professional services to the industries, most notably the assurance schemes, but also we have a feed advisor register, which ensures people offering nutritional advice on farm are adequately trained and qualified. And we also have a forage analysis assurance group, which runs proficiency testing system for labs carrying out forage analysis. Next slide, please. So really what we're about, our guiding pillars are competitiveness, productivity, and the environment. And reason for putting this slide in, other than that keeps my boss happy, is that it's really these pillars that made the PathSafe project so interesting for us, because we, as an industry, the feed industry is all about competitiveness, productivity, and the environment. Our role is to provide optimum nutrition to livestock to ensure that they are productive. Ideally, their farmers are profitable, whilst also preserving the environment. And one of the areas that is of concern to the feed industry is the is AMR and Salmonella in particular, and we want to help with the research if at all possible. So next slide, please. So what do? We lobby and represent our members. Just to be clear, that's not the kind of lobbying where we have international expensive paid trips for MPs. It tends to be, please come and let us talk to you, and you might get an invite to our conference. We hope to try and shape policy to ensure that the aims of legislation are achieved, but also that our industry remains productive and competitive. To provide information onto our members, trade assurance schemes that I've already mentioned, and also to offer training and technical support to our members. So next slide, please. So talking a lot about our members, these figures are quite old now. They're around about 5 years old. We're just doing a new survey to update this, but we have around about 230 member companies. In terms of tonnage across our sectors, we believe that we have well over 95% of the market in our association, and the value was, let's say 5 years ago, around about £9 billion of GDP. We believe, looking at the figures coming in our new survey, that's going to be probably 50% higher, sort of somewhere around about 14 billion. Well over 40,000 direct and indirect employees. And one of the things that we keep emphasising to our members and to anyone else that will listen, frankly, is that we are a key part of the food and drink supply chain. And also all those other public goods like the environment, like food security. So yeah, we think it's important that we have a voice. So next slide, please. So animal feed priorities. We're really looking, always looking at alternative proteins. We're obviously aware of the sustainability issues, deforestation around soy and palm. We've been key with feeding the circular economy, such as the UK FFPA that we mentioned before. Particularly at the moment, we are, as GB, since Brexit, we're very focused on additive and medicated feed authorizations within GB, and ensuring that they, as far as possible, keep pace with the European Union. And then on the feed safety side, we're again making sure that we keep in touch with the FSA and make sure the regulation remains fit for purpose, and that we continue to work with assurance schemes abroad, because the UK feed industry doesn't operate in isolation. One thing to say, if the slides will be circulated afterwards, there's a few links in there for various animations explaining the work of the sectors and the organisation as a whole. They're only around about two minutes long each, so that hopefully they'd be useful for you to understand what we're about. Next slide, please. So why did we why did we get involved? We were initially contacted by the VMD, who we have ongoing contact with on veterinary medicines in feed, which is one of the routes for delivery of veterinary medicines including antimicrobials into farmed livestock. Although the sort of discussion by Eamon earlier on dairy is probably the one sector where feed isn't the main delivery method. So that is an entirely separate area. And initially we went into a discussion to understand what the scope of the project, and also to arrange a feed mill visit to allow the organisers of the project to understand the practicalities of what a feed mill looks like, and what was possible and what may not be possible. So that was really my role in it. I feel quite a fraud sitting here and talking about it, but I just introduced a few people. So next slide, please. So it was actually a really tricky decision on whether we wanted to be involved, because as an industry, the feed industry takes its responsibility around AMR seriously, but it does sometimes feel like we're an easy target, a scapegoat for NGOs that actually, you know, we only use antibiotics when they're prescribed by a vet who was diagnosed an illness in livestock. So we don't actively use antimicrobials, and haven't done for 20 plus years in as growth promoters. And we also feel that maybe salmonella is also another area where the feed industry maybe has a higher profile than it deserves, in that when we review the salmonella results, we do a lot of testing. We maybe get 1% of salmonella samples a year are positive, and very rarely do the serotypes align with what is found in food and food producing animals. So it's we take our role seriously, but we are not the only answer. So that was part of the thinking for wanting to be involved. But we also felt that, having said all that, if we are introducing exotic AMR onto farms via feed, then we want to know if we're not implicated, if the results say actually we haven't found any of this AMR in the feed, then that's good news. You know, it's a gamble, but there is a possible risk to the reputation if there's a real problem. In that, you know, it's already been mentioned that these projects, there's been some research in Sweden and the USA. We could be the first country in the world that really identifies that there is a risk here, at which point UK feed industry, UK livestock industry gets some bad press, when actually we're just the ones that know about it, rather than the being in ignorance. But the confidence that in general salmonella is relatively low level in feed meant that we felt that the chances of that being antimicrobial resistant salmonella was also pretty low. Next slide. So in terms of the project, we identified a single site, predominantly for convenience for the to get the samples to the APHA lab for the analysis, but also to take to use a site that was producing a range of feeds for different species, using a fairly typical range of feed materials coming into the site. The site has a mixture of heat treated and non-heat treated feeds. When we talk about heat treated, we're talking about a process that is designed and managed to achieve a salmonella kill step for poultry feed. Even when we talk about non-heat treated feeds, there is some level of heat, but it isn't a validated kill step. So heat treatment would typically be 74 to 80 degrees C for 2 to 5 minutes. Non-treated feeds, pig feeds, for example, would probably be around about 60, 65 degrees, but then would also, whilst at that temperature, be then subjected to pressure through the pellet press, which there is some evidence of that not being very good for bacteria, but it's not a validated kill step. So really the whole process is to take a range of feed materials based on them being the most commonly used and the highest perceived salmonella risk. So that would typically be oilseed byproducts, the meal from soya oil, rapeseed oil production, and then yeah, a lot of those will be UK sourced, but also the imported, particularly the soya. And then yeah, so the samples have been taken and been tested, and I don't have any information really on the results. I believe that that there's not much sign of anything worrying at the moment. Next slide. Okay, so that's everything from me. So I will now hand you over to Manal, who will talk a little bit more about the project.
Thank you, Simon. Okay, so you've heard about how the whys of why Simon and Amon's organisations have got involved in PathSafe, and so I'm just going to go over some of the logistics to doing the testing of the milk and animal feed. Next, please. Okay, so this is just an overview of all the various aspects of the project. So at the beginning, obviously, the study design, where the agreement has to be put in place with farmers or the feed mill. The planning of the samples and the coordination of samples to the various laboratories. And all this came through via APHA, and we also supplied the collection vessels for both the feed mill and the milk. So I'll go into the individual details for the feed and the milk side of things, because they were slightly different. So the microbiology for the feed happened at APHA Newcastle, so they targeted various bacteria and froze these, and the milk went to APHA Starcross to do the same thing. Then all the samples went to Starcross for the AMR phenotyping, so the resistance phenotyping, which I'll explain in a bit. And then all the samples went to APHA Weybridge. First, we did the moldy, so identification of Enterococcus species, because we're interested in 2 species, so Faecalis and Faecium, and that's where the moldy came in. And then all the target bacteria were sequenced using Illumina, and I'll show you that in a bit. And then finally, there's whole genome sequencing analysis and the epidemiology analysis, which I'll explain in a bit. So can we go to next, please? So first of all, feed, as Simon just mentioned, so the feed mill staff are the ones that collected the samples for us. Initially, APHA staff went down to train them how to avoid cross-contamination between the different samples. And on the right-hand side, you can see the different raw ingredients that were sampled, as well as the finished products. So then mill staff collected and sent 25 grams of each of the sample. In the bottom right, you can see the bags of the various samples that were sent. And so this was done every week, where 10 different raw ingredients and 10 different finished products were sent. So in total, we're looking at 300 of each type of product. So then the samples were couriered to APHA Newcastle, usually on a Wednesday, for the microbiology to start, and this has happened between January and is coming to an end in a couple of weeks. So next, please. So in terms of the microbiology, that was done at Newcastle. So 25 grams of each product was placed into enrichment broth, and so Enterococcus, Klebsiella, E. coli, and then the extended spectrum cephalosporin resistant bacteria and the carbapenem producing bacteria were enriched in brain heart infusion, while Salmonella enterica was enriched in buffered berbertone water. So on the left-hand side, you can see the target bacteria we were after. So the E. coli, Klebsiella, and Salmonella and Enterococcus, were from non-selective plates. And then we've got the selective plates being the extended spectrum cephalosporins and the carbapenemases. And at the bottom, you can see some examples of what the colonies look like and the expertise needed to pick the colonies. So the first 2 on the left are different species of Enterococcus. So there's a Faecium and there's a Faecalis in the middle. And then there's also the carbapenemase producing E. coli. So that was a split plate that identifies carbapenem and oxo-producing bacteria. So following selection on the target's plate, a colony was taken and then purified twice on blood agar for freezing, and then subsequent testing at Starcross or at Weybridge. So the whole process from start to finish takes about a week. Next, please. Okay, so then when we think about the milk PathSafe project. So as Eamon said at the beginning, they got the agreements from farmers to be included in the study, and LLM coordinated the farm selection. So they also collected the milk sample. So weekly, Starcross would get about 25 milk samples for microbiology testing, and then the same sample at 1 mil would be saved for the integron PCR that Chris mentioned at the beginning. And these were usually sent frozen once a month. Okay, and then so the samples for milk usually arrived on a Friday, refrigerated over the weekend, and then microbiology testing started on a Monday, and that usually took about a week as well. And yeah, so the testing has been going on since January and is due to complete in a couple of weeks' time. Next. Thank you. So the microbiology side of things. So APHA Starcross took 1 mil of sample from the 5 mils that were given, and these were direct plated onto selective agar for the various different bacteria. And then the same sample was put into enrichment broth, so either buffered peptone water, brain heart infusion for Enterococcus, or Miller-Hinton broth for MRSA, and then plated onto selective media for the target bug, sorry. So, and then here you've got some examples of the plates that we're looking at. So on the right-hand side, you've got the McConkey, which is nice and easy to see the difference. So that's just E. coli on that plate. And then on the blood plate on the right-hand side, you can see there's several different colony morphologies, but really we're only after one type. And there's about 2 o'clock, there's like a halo of hemolysis, and that's the target Streptococcus that we were after. So so once that colony has been identified, then that would be purified until you get a pure colony on a blood plate and then frozen. And again, the process took about a week for the set of samples. And next. Okay, so then both types, so both the milk and the feed, the target bacteria had minimal inhibitory concentration testing to work out the levels of resistance that they had. So there's a commercially available system from Kingfisher where plates are inoculated with the different bacteria, different antibiotics at different concentrations. And in the lab, we add the bacteria on the little. On the right-hand side, you see the first machine on the left of that picture where you inoculate the bacteria into the plate at a known concentration. You wait 24 hours after the incubation, and then that gets read into like a camera, and then you can work out the MIC level. And then we've used epidemiological cutoffs to determine whether a bacteria is susceptible or not susceptible to a given antibiotic. And next. And then the samples at Weybridge. So as I said before, the Enterococcus is pretty hard to tell the difference on the plate between the Faecium and Faecalis that we were targeting. So MALDI is used for that, where you're looking to compare, you're looking at the protein profile of the bacteria to say whether it's a species, the target species or not. And then all of the target bacteria are whole genome sequenced, so they're extracted here at Weybridge using Kingfisher, sorry, I got the other one wrong, didn't I? Kingfisher, and then Illumina sequence here at Weybridge. And then the data, we use that to identify the species of the bacteria. And then we're also looking for the AMR genes that they harbour and any plasmid replicons. And where possible, we will do some genotype-phenotype correlation to see that whole genome sequencing is able to determine whether something is resistant or not. And then where time is permitting, we'll for some target organisms, we will do some whole genome comparisons to look at the epidemiology. Okay, and next, please. so the final one. So this is then we have all this AMR genotype and phenotype data, and then the epidemiologists come in, where they look at the data that we've generated, as well as the information we've got from both the feed mill and LNM. So with regards to feed, as has already been mentioned by Simon, the products are selected because based on volume of the raw and finished ingredients produced by the mill. And the aim of the epidemiology is to determine the proportion of AMR within the ingredients or the finished products and estimate the prevalence of pathogens within the samples. And likewise with milk, so as Eamon mentioned, so farms were selected based on regions within Great Britain, and each region, representative sample was taken proportional to the size. So you see on the right-hand side, the darker purple means there's more cattle in that area. And then, so the aims of the bulk milk was to estimate the prevalence of resistant bacteria and AMR genes in the cattle population, and then also compare any regional differences between AMR resistant bacteria. And then finally, thank you, the acknowledgement. So there's, as you see, there's lots of people involved at various different stages. So we've got the bulk, the National Milk Laboratories, as well as the AIC involved in collecting the samples for us, then Starcross performing the MIC analysis for both milk and feed, as well as the microbiology for milk. Newcastle did the feed samples and all the microbiology. Weybridge, James, John, and Natasha and Graham are doing the Mouldy and the whole genome sequencing. Then we've got the epidemiologists, so Chloe and Martina, and obviously lab testing, who are doing the sequencing for us. And we've got Chris and Martina and various others at APHA involved in the setting up the project. And obviously Veterinary Medicines Directorate and Food Standards Agency for support. And I'll pass over to Ed, I think.
Thank you very much, Manal, and thank you all for that talk. Really interesting presentations from all of you, and really great example of collaboration within PathSafe among different organisations in different sectors. So yeah, I thought that was great. Really enjoyed that. Very close time, I think. I can't see any questions in the Q&A. Then we might have time for one extremely quick question if anybody has something quick that they want to ask with a raised hand. If not, and a question occurs to you afterwards, please feel free to email pathsafe@food.gov.uk and we can forward that question to the most appropriate person. Happy to do that after the meeting is over. So, assuming there are no questions, just remains to thank all of our presenters for really great and interesting presentations. Thank you all for coming to join us and to watch. Reminder that we do have more PathSafe webinars coming up, and the next one in October will be a presentation about the PathSafe process evaluation, so not to be missed. So I think at that, bang on 3 o'clock, we will close the meeting, and thank you all once again.
Thank you. Bye.